what are your current position and field of interest? Could you please describe the group you lead, its facilities and technologies? What are the main objectives of the group, and what kind of research activities are currently ongoing? What are the perspectives for future research in the biomolecular field over the next years? And how might artificial intelligence impact research? What advances are expected, and how could they contribute to the development of new technologies and research approaches in this field? And now we move to Professor Reinhard Büttner. Rainer, please.
Yeah, hello everyone. My name is Reinhard Büttner. I'm a pathologist, diagnostic pathologist, just as Umberto. And I'm the director of the Institute of Pathology at the University of Cologne in Germany. Cologne is one of the founding institutes for that were referred as excellence centers in oncology, which is a program that was funded by the German Cancer Aid starting in 2008. And we named ourselves a CIO Center for Integrative Oncology. The reason for that is because the initial founding idea was that in the center of oncology is the patient. And around the patient, all the disciplines, the analytical, the therapeutic, the disciplines have to assemble and find a personalized concept which fits and is adjusted to the individual patient's need. Cologne is also part of the National Center for Tumor Diseases, which is a federal funded program to encourage early clinical research and trials. And here again, pathology is an important tool to enable early clinical trials in a sense that eligible patients fit a specific therapies and that can be tested in a clinical trial. So the interest I have is, of course, to enhance the capability of diagnostic pathology in order to come to a better and more comprehensive diagnosis, and also to apply molecular diagnostic tests and molecular pathology for diagnostics, and to merge these informations with artificial intelligence-driven analytical tools that we can easily also apply on tissues and images. And a third very important component is by having a deep look in a tumor, we of course find patients that are at risk to develop tumors because they have a hereditary trait that make them risk families. And that is also an important task of pathology, not to miss this information during a diagnostic pathology. In research, my group is trying to build animal models where we can, as closely as possible, mimic therapies that will be applied to human tumors, and thereby understand what are the critical factors that shape response of tumors to therapies, but also would allow to predict the outcome. Give you one example, tumors of the upper GI tract and esophageal cancer is the major issue here in our surgical oncology center. These patients typically receive neoadjuvant, very intensive radiochemotherapy, then undergo resection. So we have a pre-therapeutic and a resection specimen. And the extent of response as measured from the resected specimens mainly determines the outcome of the patient. So we have tried to do this with tumors in mice that resemble genetically the human adenocarcinomas, and also then find genomic, and transcriptomic, and proteomic signatures that would correlate with the response and the survival of tumor cells, and then went back to human samples where we found that the activation of these survival and rescue signatures would allow us to predict that a patient does not benefit from such toxic and intensive combined chemotherapy, radiochemotherapy. So this will be, I think, a concept that is applied to many tumors to find out patients that will not benefit from our current therapies, to spare and save toxicity, but also to find the critical vulnerabilities of tumors where we can intensively hit and target a tumor where a tumor is dependent, let's say, on a pathway or a mutational pattern or an immunological pattern. With that, I was one of the founding members of what we now call the National Network Genomic Medicine Lung Cancer. And lung cancer per se is the paradigmatic tumors where we have a lot of different therapeutic abilities, from targeted therapies to immune therapies to combined immune chemotherapies, now with the advent of metabolomically driven therapies. So an oncologist that sees a patient with an advanced stage lung cancer it really has to make a number of choices, probably more than 30 choices of what he could do with that patient. And that depends on a very deep look at immunohistochemical, molecular, and transcriptomic features of that tumors. So we apply in my institution everything from deep sequencing with Novasec technology to transcriptomic technologies, but also targeted transcriptomes like a nanostring technology, to multiplexed immunohistochemistry, to methylomic analysis, but also good old histology, which is assisted with artificial intelligence algorithm, a very, very good biomarker per se as well. And here I would like to give you another example. We are the central hub of artificial intelligence driven histopathology in Germany that aims to build up a national archive of images from the most frequent solid cancers. Several years ago, we realized that with a computerized analysis of an image, you can immediately see whether a carcinoma results from an HPV infection. Let's say in the oropharyngeal squamous cell carcinomas, we have the HPV-driven tumors, which are clinically, epidemiologically, and also with respect to therapy different from the non-HPV tumors. Now, and the features that you can extract with artificial intelligence is the kind of immune interactions from the tumor micromilieu to the tumor cells proper. And that is different in HPV positive tumors from HPV non-positive tumors. Now, the interesting thing is that some of the tumors that were on molecular tests, HPV negative, looked in the image analysis very similar to the HPV positive tumors, and vice versa. So you can now build an HPV-ness score and sort of classify a tumor. Would it behave like an HPV positive tumor, independent from the result of a molecular test? And that is now being tested in a clinical trial, whether these, let's say, more immunologically controlled and driven tumors have a different outcome under reduced therapies. So to summarize that, I think the vision, let me put it that way, I'm in pathology now for 40 years. And when I started, let's say lymphoma pathology, for example, as you know, Stefano, was in its childhood shoes. Yeah, it just started to classify large cell, small cell, and whatever. And Ricardo will also remember that. And now, I think the main issue still to classify on a biopsy in a very precise and correct fashion the tumor. But the more challenging question is to derive a deep biomarker analysis in order to come away from the traditional classes, find the individual needs for that patient. So that will require genomic testing, let's say by tissue, extracted DNAs or RNA transcriptomic features could be done also in part of blood. It could also be extracted from images with artificial intelligence, may it be histology or functional radiology. We have to extract immunological features to understand better tumors that are responsive to immune checkpoint inhibitor therapies. And we also will extract metabolomic features because some tumors are highly dependent on metabolites from the Krebs cycle. And that offers a new possibility for some of the very difficult to treat tumors because they may be dependent on a few metabolomic metabolites. With that, I think pathology has moved from a mere, let's say, histopathological lab science to a clinical science. It has arrived much closer to the patient and to the therapeutic decisions with the patients. And the last point I would like to stress is that that was said by the previous speakers very precisely in much better words than I could do it. But when you diagnose a tumor, that tumor has come a long travel and has made interactions with the tumor microenvironment. It may have a mutation, a driving mutation. It may have several mutations, may have no mutation. We can explain, but it has has formed a microorganism, and then we take a single biopsy, and then the patient will travel another period of time. And then maybe in some instances we take a second, in some instances the third biopsy, and we monitor that disease over time. But it will be in pathology extremely important to have prognostic, predictive algorithms that would tell us what will happen in the next year or the next 2 years with that tumor under therapies. And that is the challenge I think we are currently working on. Thank you.
Thank you. I fully agree. I'm a pathologist, so I can only agree on the present view of pathology, which is indeed different from the one which we were familiar with at the beginning of our career. Knowing quite well the organization in Germany, because of the fact that I spent about 2 years of my life in Germany, I think that there is a very important point, the organization in Germany. So the fact that there are reference centers which allow the centralization of samples, techniques, and expertise. I think that this is a very important point. Am I correct?
Yes, and I can, of course, you allude to the reference centers for lymphoma pathology, but I may give you a more recent example, I think, which illustrates the importance that at least once every time a patient is seen in a center where all specialists come together and make a therapeutic concept. As I told you, we have founded the National Network Genomic Medicine Lung Cancer. That is organized in 25 national centers that have built molecular path labs that diagnose lung cancer samples in a very synergistic way. So and then you have thoracic surgeons, pneumologists, oncologists, radio-oncologists, and so forth. and they come to a therapeutic plan. And that plan travels with the patient. The patient may be treated by a local office-based oncologist far away from these centers. And recently, the government has reevaluated that concept and asked if we put money in that, does it improve anything? And for me, the surprising result was, and that is currently under publication, is that if you take matched patients, same age, same tumor stage, same histology, treated inside the network or outside the network, the difference in progression-free survival and overall survival is massively, significantly different, with a hazard ratio of 1.72. And they all receive the same drugs. They have all the same possibilities, that their healthcare pays them the same way, but it's coming together to concentrate interdisciplinary analysis and coming to harmonized therapeutic plans. I think that is something important for all of oncology, for all tumors and all of oncology. And the interesting thing is that the evaluation was also in an economic way, and the patients that are diagnosed and treated inside the network spend less days in hospitals, meaning that they probably compensate for the additional therapeutic costs. So it's economically beneficial, but it's also, it works like a new drug. It has a clinical trial. The hazard ratio is really surprisingly.
Stefano, can I bounce on that for a very small question?
Yes.
Yeah, Reinhard, I was very interested about you describing your hub in artificial intelligence assisted pathology, because I know that's one of the great difficulty in this application is the interoperability, and just the staining is a huge problem if it's done differently in different places and different institutions. And I would like you to allude a little bit on how you went over this. Did you do segmentation? How did you do that to go over this very basic problem of intelligence artificial based analysis?
Yeah, a very important question, and something that also you should invite our director of virtual pathology, who's a Russian resident, also an excellent uropathologist. But I think there are two critical factors in the game. First, and that came to a surprise to many of us, is the quality of the original slide. Because in pathology, we have thick slides, thin slides, we have artifacts, we have dust particles, we have an H&E is not an H&E. So we have huge differences from lab to lab, inter-lab variants. And we published recently, in the last year, a paper in Modern Pathology where we describe precisely, we have modeled precisely the influence of these histological artifacts on the slide and their impact they will make on the AI algorithms. So the AI algorithms massively deteriorate when the quality is low. So that is, you need better histology for computerized images rather than poor images. And the other thing is, of course, segmentation is key. If you don't train and segment your algorithm to what you would like to see, then it's very difficult to control. That is something that is the first step, to train and segment, let's say, a set of classical images. However, that carries another problem, because, for example, URI has just developed and published an algorithm for colorectal cancer detection. which is quite easy, I would say. But then you have outlier tumors that look very different, like microsatellite unstable tumors. They can look like lymphomas. And when they are so much outside to what you have trained and segmented initially, then they will always be a problem to the algorithms. And then you have to sort of integrate your outliers, improve. So AI is not something you buy from Google, switch on electricity and it works. It will accompany you lifelong, as much as a pathologist will learn lifelong. So we have to assemble larger and larger data sets to get better and better. And the difference between 99.5 and 99.8% specificity is much more difficult than the one between 50 and 60%.
I feel agree. The problem of the pre-analytical phase, as well as the difference, if we focus on the HD, there are at least 300 different preparations. So it's not easy to apply the artificial intelligence criteria to the histopathological routine on a daily basis.
That is something you can deal with, because when we do segmentation, we not only segment anatomical regions within the tumor, but we also would like to see slides from the US, slides from Austria, slides from Germany, slides from Russia. So to cover the entire image spectrum that will be looked at with, in later on.
I thank you very much for taking part, and I'm looking forward to meeting you all here in EAG. Bye.